where she studies Mexican Americans in Bronzeville, a population with high rates of obesity and poorly controlled diabetes. Her work identified chronic inflammation and impaired immune responses as key factors increasing susceptibility to lower respiratory tract infections. Since coming back to Pakistan in 2015, she has led various national level cohort studies on immune dysfunction in people with different comorbidities like diabetes, and also worked on pneumococcal disease burden and vaccine effectiveness in children and the elderly. And she is also part of the Wellcome Sanger and the Gates Foundation funded Global Pneumococcal Sequencing Consortium. And she's going to be talking about today about the Great Escape, capsule replacement, capsule switching, and vaccine evasion, the current situation and future directions. Thank you, and over to you, Dr. Shaper.
Thank you so much, Dr. Imran, for such a generous introduction. And happy to present here today. And again, congratulations on having such a successful workshop. I wish I was there and taking part in everything with all the other participants, but maybe next time. Better luck next time. So like Dr. Imran said, that the topic of my talk today, or my discussion, and in fact, we can make it very interactive. So if you guys have any questions, raise your hand, stop me in the middle, and I'll be happy to answer your questions. The Great Escape, and there is a reason behind choosing this title, and the most important reason is the remarkable ability of pneumococcus to change as you try to eliminate pneumococcus. So it's shifting shapes, it's changing shapes, it's changing its epidemiology, the population biology of pneumococcus is changing. And you have already experienced that in the last few years, since we have started vaccinating children with pneumococcal conjugate vaccine, we are learning more and more about pneumococci. Now, the task here is how to tame this dragon and how to prevent this bacteria from constantly evading the vaccines and constantly evading therapy and evading our immune system. So I'm going to take you guys through different aspects of pneumococci, and we will start with simple biology of pneumococci. And I know I'm going to be preaching the choir, and I know you already know a lot about pneumococcal biology, but there are some interesting facts in pneumococcal biology that I think we should all know, because this gives us more appreciation of the bacteria. And then I'm going to take you into a little bit of epidemiology of the infection. Since we had PCV vaccine, since we started administering PCV vaccine, how the global picture of pneumococcal infection started to change, how pneumococci started to evolve, then what we have done in our lab, and how things started to differ when we started with PCV10, then we moved over to PCV13, and then what lies ahead? How are we going to, like I said, tame the dragon? Are there any ways out? Are there any other antigens? Are there any other targets that we can use to make vaccine? So just stay with me, bear with me, and like I said, wherever I lose you, just let me know, and I'll be more than happy to take you through it. So this is the disclosure statement, and I declare I don't have any financial relationships with proprietary entities producing, marketing, reselling, or distributing healthcare goods or services consumed or used in patients in the past 24 months. I wish I did, but I don't. So here we go. So in case of Streptococcus pneumoniae, like I said, that it's a shapeshifter. And all those people who are working with pneumococci and have looked at pneumococci very closely, I'm sure you have started to appreciate the fact how pneumococci shift shapes. So it goes from one serotype to another serotype, from one genotype to another genotype. And then now we are mixing all of those things in global pneumococcal clusters. And those clusters are also bursting out of their seams. And that's just because of the ability of this teeny tiny bacteria to evolve. And as we go along, I'm going to talk about the mechanisms, two different mechanisms of evolution within pneumococci. So if you look at pneumococci, most of the time you call it it a diplococci. So it's two bacteria which are joined by impartially hydrolyzed cell walls. So, there is autolysin that is responsible for hydrolyzing the cell wall, and during cell division, lots of different things happen, but sometimes these two bacteria don't get to separate, and that's why it remains as diplococci. Now, in most of books, pneumococci is stated as diplococci. When you look under the microscope, not all pneumococci are going to look diplococci. Like serotype 4 will look like a diplococci. It's a characteristic diplococci, typical diplococci. But if you look at serotype 1, sometimes you will find two bacteria together. Sometimes you're going to find 3 bacteria together, 4 bacteria. And if you view the late log phase culture, you can actually see very long, beautiful chains of pneumococci. So, and that's also pneumococci. Primarily, it is found in nasopharyngeal cavity. And, if you don't do anything to pneumococci, it doesn't harm you. It can sit there, and it can sit there from weeks to months to years without causing any infection. And what it does is that it invades your superficial epithelial cells, and then it comfortably sits there unless something happens to you. So in children, most of the time, and a lot of papers have been published. If you review papers from Jeffrey Weiser's group, Jeffrey Weiser has been a champion in carriage studies, and he had very convincingly showed that experimental human carriage can continue for months and nothing happens to these people. But if you have a superadded infection, like a real infection, and this infection could be with influenza A virus, it could be Haemophilus, but these bacteria and viruses actually paves way for pneumococci to transit from nasopharyngeal cavity into the lungs. And then once it's in the lungs, sky's the limit. It can go wherever it wants to. So it can then invade most of the sterile sites. So here's just a cartoon of carriage going into transmission and invasion. So basically, this is what I was trying to say. That you see, you have basic epithelial cells, a little bit of mucus. Pneumococci is going to hang in this mucus for a little while, but then pretty soon it is going to break itself out of the mucus. It will reach these epithelial cells, and it's not going to hurt you too much. It is just going to dig a little bit deeper into these epithelial cells, and it will just stay there. Now, a lot of people might think that it forms biofilms. Does it form biofilms? We don't know. It may form biofilms. So if you want to know about pneumococci and biofilms, you should read papers by Edward Sword. Now, Ed Sword, what he did is that he did a co-infection. So he infected chinchillas, and the reason why he used chinchillas is because the anatomy of the inner ear of chinchilla looks very similar to young children or to infants. And he gave chinchillas both Haemophilus and pneumococci together. And there he showed that, yes, pneumococci has the ability to form biofilms, because for years, the literature was contemplating whether pneumococci forms a biofilm or not. However, whether pneumococci forms a biofilm in nasopharyngeal cavity during carriage is still a debate. So if you have funding, you can always go and explore and figure out whether pneumococci forms a biofilm during carriage or not. But the long story short, once it enters into your nasal epithelium, it can either sit there comfortably without doing anything. But if you have any other infection, it can be shed into mucus, into your cough droplets. It can hang in cough droplets. And then another interesting thing about pneumococci is that other than human, there are no known hosts of pneumococci. So human is the only known host. Fomites, yes, it can survive on fomites. It can persist on fomites for weeks. But see, pneumococci is very delicate, and it is very susceptible to drying because it cannot form spores. All it has to coat itself is the sugar-coated capsule. So that's why don't expect pneumococci to survive on fomites for a very long period of time. Okay. Achha, now, once it starts entering into the host, because of anything, your immune system went down, you had another infection, you had another bacterial or a viral infection, then that's the time when pneumococci is going to invade. And once it invades, it can, through aspiration or through any other bacterial or viral infection, it can enter into your lung and cause pneumonia. It can also be loose in blood, and from blood, it can enter into your brain, cause meningitis. There could be some local spread of infection, and it can enter into your middle ear, causing otitis media. Now, here's one more thing that I would like to add. Most of the data says that bacteria enters into your brain through hematological routes, but there is a lot of data that shows that non-hematogenous routes can also take pneumococci into your brain. And how it works is that pneumococci can climb through your nasal bulbs and it can then enter into your brain. It doesn't have to come into the blood because, see, once it enters into the blood, there is complement, there is antibodies, and there is neutrophils. The three things that are kind of toxic or detrimental for extracellular pathogens. So they are going to kill pneumococci. So it tries to find routes through which it can enter into other spaces without being detected by the immune system, without being killed by the immune system. So see how smart it is. Anyway, moving on, I wanted to, so I don't want to bore you, but this is really important. I want to talk just a tidbit about the host pathogen interaction and pneumococci. So here I got the slide, and don't get confused in the details. All I wanted you to appreciate here is that this tiny bacteria has few virulence factors. In fact, it has a host of virulence factors, which can be both outside the cell or inside the cell. So intracellular and extracellular virulence factors. Among them, one of the most important one is PSPA. Then it has the pilus, which is also called RRG. Then it has PSPC. And using these 3 or 4 virulence factors, it stably colonizes your nasopharyngeal cavity. Now here you see it can bind to the polymeric immunoglobulin receptor here. And you know, binding of PSPC to polymeric immunoglobulin receptor also allows for the movement of pneumococci from basolateral to the apical side of the cell. And this is exactly, so it is just basically piggyback riding on polymeric immunoglobulin receptor, which is sitting out there for binding of immunoglobulin A or IgA. Then it has neuraminidase. Neuraminidase is also found in viruses. So if the virus comes first, then it's going to use its neuraminidase in order to cleave most of the mucin, and that is going to facilitate movement of pneumococci from nasopharyngeal cavity into the lungs. Now, PSPA is kind of an interesting protein. PSPA is like capsule. PSPA is always present on pneumococci, and PSPA is found on all pneumococci that has been isolated to date. PSPA is responsible for preventing complement deposition. It is also responsible for inhibiting phagocytosis. It also binds to the mucosal proteins like lactoferrin and prevents killing of bacteria by lactoferrin. So it's a multifunctional protein. It's immunogenic also. And PSPA is actually antibodies to PSPA cross protects. And then basically the rest of it here is the same that I just talked about, but the most important or interesting thing is the immune evasion. So see, once it enters into the blood, then it is going to secrete so many things. The most important thing that it does is that it has PSPC. So now in the presence of capsule, PSPA and PSPC complement, it's difficult for the complement to deposit. So if you have IgG, then IgG can absorb pneumococci and it can facilitate deposition of complement, which will allow for opsin of phagocytosis. If you don't have a lot of antibodies at the time of bacterial invasion, then PSP and PSPC, they're never going to allow for complement. And you see this thing here, factor H. Factor H is the factor that belongs to the alternative complement pathway. So this smart guy is blocking both the classical and the alternative complement pathway. If you go into the blood-brain barrier here, also it binds to platelet activating factor receptor. And for that, it doesn't even need a receptor. It doesn't even need a protein. The choline, the phosphocholine, which is present in the cell wall of pneumococci, it's going to use that to bind to platelet activating factor receptor. Then you have another protein, which is pilus. Pilus is also going to bind to neurons. And then pneumolysin. Pneumolysin is an autolysin. It is activated by cholesterol. It's going to destroy the blood-brain barrier and allow for pneumococci to invade. Once it invades into the brain here, your RRG, which is your urbilin, also is going to bind to beta-actin. And once pneumococci stabilizes itself on the neuron, it's going to release pneumolysine. Pneumolysine will destroy the neuron, allow for the pneumococci to enter into the neuron, and that's the death of the neuron. So you see bacteria, this teeny tiny bacteria, I'm sure that you have already started to appreciate how this little bug is causing so much destruction in the body. And here in gray bars, I just listed a few steps. So here it is epithelial adhesion and most probably biofilm formation, inflammatory response. This is a basic mucosal inflammatory response that is going to try to minimize bacteria. But what it does to evade this is that it just enters into the epithelial cell, take refuge into the epithelial cell and allow for themselves. So they're not replicating, so immune system is not too worried about it. They're just taking refuge into these cells. If they are allowed and if the conditions of the microenvironment of the niche where they are sitting is conducive, then they are going to come out and they will start replicating. And then, of course, they are going to get transmitted into different parts of the body. And then, of course, body is going to induce inflammation. Some of it is going to be just what you call general inflammation or non-specific inflammation, which will be induced by macrophages and neutrophils because it enters into the lungs and it activates the alveolar macrophages. And then if it enters into the blood, then they have monocytes, which converts into macrophages, and they have neutrophils. So here's a little bit of host pathogen interaction. If you keep this in mind, you will know what other targets can be used for vaccine. Okay, so we move a little bit into the disease burden, and I don't want to quote any numbers because I think that numbers are at everybody's fingertips because those numbers have been reported, re-reported in multiple places, so you all know. But I found this particular graph extremely informative, and what it is trying to show, first of all, it gives you an overview of everything that has been happening since the immunization started, since PCV immunization started up to 2015. And this is where the red arrow that I made here, this is where your PCV7 administration started in Europe and USA. And what it shows that before that, you had a good 600,000 to 700,000 cases or deaths due to pneumococcal infection in 2000. And this green thing is showing proportion of children which are not immunized. So at the start of the immunization, of course, 100% of the children were not immunized. But as you go along, the number of not immunized children go down, and the deaths are going down too. Now, if you only look at this picture, this presents a very rosy picture that if you give vaccine, the deaths go down, and over a period of about 15 years, we have accomplished a lot. In part, it is true because when you give vaccine, the vaccine serotypes starts to get eliminated. Or no, I'm going to rephrase it. Some of the vaccine serotypes actually starts to get eliminated, but then some other serotypes starts to invade the niche that is now vacated by the vaccine serotypes. And it is these non-vaccine serotypes that start to cause infection. But overall, if somebody asks you whether PCV vaccine works, so the simple answer is yes, the vaccine works, and it can eliminate all the serotypes that are present in the vaccine. It can also bring the burden of infection down, which is caused by those PCV vaccine types. So going a little bit deeper into the burden, this is again data between 2000 and 2015. And here it's presenting a global picture of what's happening in the world and how pneumococcal vaccine is impacting the burden of disease. There are two very important data here. One is the mortality rate, and the mortality rate is indicated by this color. So the yellow color means that the mortality rate is sometimes somewhere between 25 to less than 100 per 100,000 individuals. The red means a little bit higher, which is more than 200. And then there are these circles, and these circles gives you the total deaths. So the larger the circle, the more the deaths are, and they have capped it at 65,000. And in India, you see that there are about 65,000 deaths compared to Pakistan, where there are like 15,000 deaths. And the reason for this is that Pakistan started immunizing children way before India did. So Pakistan started in 2012, India did it in 2017. So, you know, by the time this data was collected, India had still not started vaccinating their children. So this gives you a very beautiful overview of what's happening in the globe. And yes, the infection rates are going down, but then they depend on multiple things. One of the things is vaccine coverage, and the other thing is emergence of new non-vaccine serotypes and persistence of vaccine serotypes, which we are calling vaccine escape strains. Bacteremia, so pneumococcal infections don't just target children, they target adults also. And in case of adults, people with immune compromised issues, people with diabetes, cardiovascular disease. So globally, there are recommendations for individuals, for elderly, especially elderly with comorbid conditions, that they should be given pneumococcal vaccine. But in Pakistan, the recommendations are kind of soft. So people who have like good physicians, private practitioners, they recommend that, you know, you should get a flu shot, you should get your pneumo shot, so that you are protected from flu and you're protected from pneumonia. But there are no government recommendations, although government is moving into adult immunization, but I think we still have a little bit of time, a little bit of window before that becomes public. So if you look at adult infections, 1 in 20 older adults die who gets pneumococcal pneumonia. 1 in 6 adults or elderly die from a bloodstream infection, and 1 in 6 elderly die from meningitis. So you see, it's causing significant amount of deaths in adults and in children. Now, pneumococci is a vaccine preventable disease, and we have been talking about the vaccines all along. And PCV vaccine is not the only vaccine. And I'm sure that everybody who's sitting here attending this talk today, they must know that the first vaccine that was made in 1977 was pneumococcal polysaccharide vaccine. It was a purely polysaccharide vaccine. And what they did is that they took 23 polysaccharides, and these 23 polysaccharides were the ones that were causing 80% of the infection. They put it together and they started vaccinating people. I'm not going to say that the vaccine was not effective. The vaccine was about 65 to 50% effective in preventing invasive pneumococcal disease, but the problem was that it could not prevent against carriage, one. Two, it was not eliciting a strong, robust response in children and also in adults. In children, because polysaccharides don't usually induce a robust response in children, because children has very naive immune system. They do not respond to carbohydrate vaccines, but they do respond to protein vaccines or protein antigens. In adults, immune senescence. So their immune system was getting older, and that's why they were not responding. The third important thing about this vaccine was that because it was a purely polysaccharide vaccine, there was no memory associated with it. So in order for the immune system to build memory, it needs a little bit of protein. Carbohydrates are repetitive, and your dendritic cells usually don't like repetitive things. They don't like presenting repetitive things. And unless they present an epitope, your T cells are not going to recognize what's happening. And then, you know, memory is gone. So between pneumococcal polysaccharide vaccine and pneumococcal conjugate vaccine, one more event happened. And that was that people came up with Hib vaccine, and Hib was a conjugated vaccine, and they saw brilliant results for Hib. And then there were two hypotheses, in fact, two parallel hypotheses. One of the hypotheses was that if we lower the burden of Hib, maybe we will get rid of pneumonia, because most people believe that pneumococcal infections are a prelude, or Haemophilus infections are a prelude to pneumococcal infection, that you have to be infected with Haemophilus or an influenza virus before you get pneumococcal infection. So if they eliminate Haemophilus from that niche, pneumococci is not going to get the support that it was getting from Haemophilus in order to transit from the nasopharyngeal cavity into the lung. And the other thing was that they saw the success of conjugate vaccine. And that's how pneumococcal conjugate vaccine came about. And they decided that we are going to take the serotypes that are causing most of the infections in children, and we are going to add them in, attach them to a protein conjugate, and we'll give them to children. Now, the reason why only 7 serotypes were added at that time was because those were most commonly found serotypes that were causing invasive pneumococcal disease. Now, you see, even though nasopharyngeal carriage is the start of the infection, so something has to be given to prevent nasopharyngeal carriage in order to prevent invasive disease. But because nasopharyngeal carriage don't do anything to you, it is not until you get an invasive disease that deaths and disease start to become more obvious. So most people are trying to prevent invasive pneumococcal disease. So the first vaccine that was designed was PCV7. PCV7 target capsular polysaccharide. Now, why only polysaccharide capsule? Why only capsular polysaccharide when we already know that it is not a very good antigen? But there are lots of things that are associated with it. It is a virulence factor. It prevents complement deposition. Antibodies to capsular polysaccharides do induce opsonophagocytosis. So that's why they decided that, let's just stick to the capsular polysaccharide because it's a known antigen and we have been working with it for quite some time. Serotypes that are included in pneumococcal conjugate vaccine, you don't really need to memorize it, but I think it was a good idea to just give you a snapshot of what serotypes are included and how this vaccine had evolved over a period of time. We are now here at PCV13 in Pakistan. PCV13 was introduced in 2021. And if you look at the way the pneumococcal conjugate vaccine is moving, we are actually going back to PCV23. So in PCV20, which they are planning on administering in adults, we have pretty much the same vaccine serotypes that were originally present in PCV23. Timeline of pneumococcal vaccine. So in 2000, PCV7 was licensed for children. 2009, 10 was licensed for children. PCV13 was licensed in 2010, 2012. It was licensed for elderly. Then in 2014, PCV13 with so along with the pneumococcal polysaccharide vaccine, which was PPV. I don't call it PPSV. So it's PPV23 was started for adults who are elderly, who are older than 65 years of age. In 2019, PCV13 was given to elderly who were 65 years or older than PCV15 and PCV20, and so on and so forth. So by 2025, they have PCV for individuals who are older than 50. Now, coverage. So when I started this conversation, we talked about two things. One is the coverage of this vaccine, and two is how well the vaccine serotypes are responding to the vaccine. So in case of coverage, a lot of, and this data was somewhere between 2000 and 2019. And so it's fairly recent data, and you can see that there are still regions in the world where the coverage is still pretty low. And if you look at South Asia, which include India, Pakistan, and Bangladesh, Pakistan is doing relatively better than India. And, we can always develop hypotheses about strains moving from India to Pakistan, from Bangladesh to Pakistan because of the cross-border transport, because of the cross-border migration. So these strains can be moving across borders. But we still don't have any biological evidence or epidemiological evidence that the strains that we currently have in Pakistan, both the non-vaccine and the vaccine serotypes, have been imported from anywhere. Now, how did pneumococcal vaccine impacted the disease? There was significant reduction, 7.24% global reduction in pneumonia. Disease prevention, 175 million cases and about 624,000 deaths were prevented. And there was a decrease in mortality rates between 2000 to 2015. Mortality due to pneumococcal infections declined by 50%, which is all pretty good. I mean, it's very decent. But, whatever data I'm presenting here, it is from the vaccine serotypes. Now, if you look at it in a more realistic situation, what you see between 1998 to 2015, that yes, once PCV7 was introduced, there was a sharp decline in PCV in infections that were being caused by serotypes that belonged to PCV7. But then you see that there were non-vaccine serotypes, which is this green line, they started to creep up. And then if you look at PCV13 and non-PCV7 serotypes, the infections were. So, we need to keep an eye on these strains, on these bacteria. And there has been an ongoing debate and ongoing controversy on how to do surveillance for this shapeshifter, how to keep an eye on these emerging serotypes, on these non-vaccine serotypes. Then you see infections in adults, and the data is pretty similar that after the introduction of PCV7, the PCV7 serotypes declined significantly, but then you have other non-vaccine serotypes starting to come up. The scientists started reviewing this phenomenon, and they called it serotype replacement capsule switching. Now here, and these two terms are being used persistently, that it's a capsular switch, it's a serotype replacement. Now, pneumococci exist in serotypes, and you can group those serotypes because they are somewhat antigenically similar. And if you group them, they become serogroups. Now, not saying that nothing is happening in those serogroups. A lot of mutations are happening in those serogroups as well. And that's why those serogroups are continuing to expand. And then there are a capsular switch. Now, in capsular switch, you may still remain within the serogroup, within the same serogroup, or you may jump into something else, like 19F becoming 14, right? So this is a totally different serotype. So, and both of those phenomenons are being observed since we started administering pneumococcal vaccine. And I'm sure that this phenomenon was happening even before that, but we were not paying attention because when bacteria is present in the nasopharyngeal cavity, there is polymicrobiome or polymicrobial community, and you don't know what kind of stuff goes on when there is a polymicrobial community. Now, what is the biological basis of these two things? And I'm going to just touch base on that. Now, I'm taking this example of 20A and 20B because it's just the simplest example and I really like talking about it. So you guys don't have a choice. So here you see this is 20A. And in case of 20A, this gene, which is actually a glycosyltransferase, this was a pseudogene. So it was not doing anything. And then over a period of time, pneumococci underwent some recombination events. Now, mutations can occur in multiple ways. It can be recombination and it can be a single nucleotide mutation. Most of the single nucleotide mutations actually cause diversity. They are very important for diversity. But when you talk about these big events like serotype switching, for that recombination has to occur. Okay? The other thing, and I think you probably all know that for capsular polysaccharide, there is a locus. And in this locus, there are multiple genes, and these genes are either regulated by the same promoter or they can each have their own promoter, but they all work in sync. So just imagine that, you have a bookshelf, and bookshelf is the DNA, and you have multiple books sitting on it. Each book is a gene, but then you decided to arrange these 6 books by genre. And it could be rom-com, it could be anything. So this is a rom-com genre that we are talking about. So there are multiple genes that are sitting in a locus. They could be regulated by a single promoter. And each one of these genes or combination of these genes are performing multiple functions. And I have tried to list some of these functions like UDP, galactopyranose pathway genes, then initial transferases, polymerases, glycosyl transferases, and then flipases. Flipases are really important because most of the capsular synthesis occurs in the cytoplasm. And then there is a carrier that takes the capsule or the already synthesized subunits of the capsule to the cell wall, and this flipase actually flips it over the cell wall where it gets inserted into the cell wall. Now, when this pseudogene was replaced with a functional glycosyltransferase, you see that there was an addition of a side chain, of an alpha-1,6 side chain. And this addition of the side chain actually changed the serotype of 20A to 20B. Now, they're still staying in the same serogroup, and they can continue to expand. And a very good example of such expansion is serotype 6. We all are very familiar of serotype 6. And, you know, people have been working, I mean, if you read papers by Dr. Ashley Robinson. Ashley Robinson was probably the first one who started working on serogroup 6. And he was the first one who showed that there are clonal complexes, which means that you can group the serogroups into one group based on allelic differences in their housekeeping genes. And this is where he found that there could be several clones or clonal complexes of 6. Now, the thing that I want you to appreciate is that just by few mutations, now see here you have alpha-1,3 linkage, the linkage changed to alpha-1,4, then 1,3, and each one of these changes are imparting a different antigenicity to the capsule. So it went from 6A to 6B to 6C, 6D, and now 6F, G, and H. And the funny thing about 6A and 6B is that they are pretty similar to each other. The only thing is the glucose is actually replaced by a ramenose, and this gives it a different antigenicity. And then the other phenomena is the emergence of the new serotypes. So the new serotypes that we are seeing are 7B, 10D, 33E. I'm not saying that they emerged after or they were born after we started vaccinating. They were always there. They were always present in the niche, but because the niche was occupied by more virulent serotypes, which we are now trying to eliminate using those vaccines, these serotypes remain buried under those very virulent serotypes. But now those virulent serotypes are gone, so they have the field to themselves. And that's why now they are emerging. And the good or bad thing about them is that they can be very drug resistant, because they have been talking to each other, they have been seeing this. And I know that the drug resistance comes at a fitness cost, but that's besides the point. We're going to talk about antimicrobial resistance some other day. So then you saw that this is really a demon, and it shifts shapes. It doesn't take that long to shift shapes. So how do we keep an eye on it? And surveillance is becoming a very important thing. A lot of you guys who are listening to me out there are epidemiologists. I'm not an epidemiologist. I just work with pneumococci and I'm happy there. But you all know how important surveillance is in order to see how things are changing and what can be done, because, you know, surveillance or data from surveillance actually informs policies. So this is really important that we understand what we are dealing with. So how do we characterize pneumococci? People did lots of things for characterizing pneumococci. Previously done studies have used multi-locus sequence typing, and I just mentioned one of the studies by Dr. Ashley Robinson, who did this study where he showed that using the housekeeping genes and the allelic differences in the 6 out of the 7 genes, you can actually group them into different clonal complexes. For pneumococci, the interesting thing is that individual housekeeping genes are 10 times more likely to evolve by recombination than mutations, and then individual nucleotides are 50% more likely to evolve by mutation than recombination. And the same is true for capsular locus, that it evolves more by recombination than by mutations. So given this complexity, whole genome sequencing is likely to answer some of the questions which are pertinent to the biology of pneumococci. This takes us to some, and here I'm just trying to help you appreciate or understand how all of these things are coming together. So now with the whole genome sequencing and with the sequence type and with the serotype, you can actually start to group these things into global pneumococcal clusters. So here I can give you an example that this was a sequence type, which was 230, and by just one allelic change it can give rise to so many different sequence types. And then here there was a one allelic change, and this one accumulated another allelic change and became a different sequence type. So, and by doing that, they entered, or they resulted in two very distinct clusters. And each one of these clusters is called clonal complex. And this clonal complex is from 24F. And if you take 14 different strains of 24F, you are going to end up with something like this, where you have multiple clonal complexes. And I'm only taking one strain, one serotype, which is 24F. If you collect 18 different strains of 24F, and this is what people have done here, 65 different strains of 24F, and they have shown the blue mark here are pre-PCV13. The pink one are the clonal complexes that emerged after PCV13. And then you can also see that these are from different regions, and they are grouping separately into separate clonal complexes. So finally, we came up with some sort of scheme where we can either separate them into clonal complexes or bring clonal complexes, serotypes, and sequence types all together into global pneumococcal clusters. Global pneumococcal clusters, our sequencing clusters are more granular because they look into the entire genome, or the changes into the entire genome. And because of this granularity, the definition of antimicrobial resistance in pneumococci is also changing. Coming to Pakistan, Pakistan is ranked 4th in the world. And from here on, I'm going to present data that we have accumulated, and some part of this data was accumulated in collaboration with Dr. Imran Nisar. Dr. Imran Nisar had contributed significantly to our work. They contributed all the nasopharyngeal carriage isolates for our first study. And of course, constant discussion and lots of things over a period of time have supported our studies at LUMS. So Pakistan is ranked 4th in the world in the burden of infectious diseases by pneumococci. 2012 to 2020, 20 to 80% vaccine coverage. And the reason why it's 20 to 80% is because when you look at regional vaccine coverage, Balochistan has the lowest vaccine coverage, which is about 20%. Punjab has the highest vaccine coverage, which is 80%. And then KPK and Sindh lies somewhere in the middle. Then about 476,000 episodes of pneumonia in Pakistan, 14,000 deaths are attributed to pneumococcal infections. Coverage. Now, this was an interesting slide, and I thought I'm going to add it to my presentation. I got it from the WHO pneumococcal vaccine coverage, and here you can see there is numbers that were reported by admin, official numbers, and then there were WHO, UNICEF, and National Immunization Centers. Although the numbers are not very different, but nevertheless, there are some differences here, because here in 2023, the administration is reporting that 97% of people were vaccinated, whereas according to WHO, UNICEF, it's 86% that were vaccinated. Now, when you compare these regional or national numbers to global numbers, there is a big divide. And here you see that in 2024, globally, 67% of the world has coverage, or the coverage globally is 67%. And now this also includes countries that recently started immunizing. This also include countries where there is a lot of migration, and then climate change, and all of these things, conflicts, and all of these things are actually contributing to the delivery of vaccine to children. For our study, the sample size for carriage strains were AKU Sindh. Then for the invasive isolates, we relied heavily on Chuktai Laboratory, and for some of the carriage isolates from Punjab, we went to the Immunization Center in Children's Hospital. They were generous enough to provide us with sample. In pre-PCV13, we had a total number of sample of 190. In post-PCV13, it's 120. And here is some distribution that the samples were isolated, or the bacteria was isolated from nasopharyngeal swabs, blood, sputum, pus, CSF, ear swab, pleural fluid, and in other places. There were decent number of samples from kids who were younger than 5 years of age and adults who were older than 5 years. Male and female were equally kind of represented. And in terms of the provincial representation, primarily we had samples from Punjab, and then we had some samples from Sindh, KPK, and Azad Jammu and Kashmir. When we look at the serotype distribution in pre-post vaccine era, and in this slide, I'm primarily talking about PCV10. Let me explain the, so that you are oriented with the way the data is presented here. In this first half, we are presenting overall numbers. The blue bars here represent samples that were collected pre-PCV10. The orange bar represents samples collected post-PCV10. And below on the x-axis, we have listed all of our serotypes. So overall, when you look at it, you can see that serotype 9V was most prevalent overall. And then in post-PCV10, 9V went down. 1 persisted, not a big difference overall. And then 19F persisted, 14 is still there. And then we have a huge variety of non-vaccine serotypes. Yes, there are some singleton strains in here, but nevertheless, they are there. But when you start to distribute it by disease and nasopharyngeal carriage, you see that 9V kind of is more associated with nasopharyngeal carriage and less associated with disease. Then the same is true for serotype 1. Serotype 1 is highly associated with disease, but you don't find it in carriage. The 19F is true that primarily it is associated. And when I say disease, I'm talking about invasive pneumococcal disease. 18C is associated with primarily nasopharyngeal carriage. And then again, in both in nasopharyngeal carriage and disease, they have their own set of serotypes that are predominantly present or associated with invasive pneumococcal disease. Then serotypes, we are trying to go more granular in this case. Serotypes over source of isolate age and gender between 2016 and 18. This is also post PCV10 in Pakistan. The two things that I want you to, or that I have highlighted also with asterisks, is the significant difference between the sources. So here, serotype 1 and 19F is primarily associated with disease. And then here you have serotype 9L, which is also primarily associated with disease. When you look at the age distribution of these, 9L is basically present. So I missed out on one more thing here. So the blue bars are actually nasopharyngeal carriage, whereas the green bars are disease. So these two are both disease samples, whereas the blue one is carriage sample. So these two samples are associated primarily with invasive pneumococcal disease. The blue bar is with nasopharyngeal carriage. When you look at the age distribution, most of the nasopharyngeal carriage is associated with children, whereas the disease sample is associated with both adults and children, but primarily with adults. And then again, when you look at the serotypes here, you see both genders are equally represented in all of these serotypes. And again, this is post-PCV10. And then again, something to appreciate here is the non-vaccine serotypes, where 6A is predominant, and then 10A, 9, and 24. And then again, there are some singletons here. When you look at the serotype distribution among the common GPSCs pre-post PCV10, so here are GPSC 5 too. The only thing that I would like to bring your attention to is that some GPSCs are primarily associated with a single serotype, and here this serotype is 15B and C. Here also you see that the GPSC2, both pre and post, is associated with a single serotype, and this serotype is serotype 1. So one thing that's becoming more common is that serotype 1, serotype 3, they are not going anywhere. For serotype 3, there is a reason, because serotype 3 does not associate its capsule with its cell wall. So the capsule is synthesized and secreted out, but it remains close to the bacteria because of the so it's non-covalent bonding, basically. So electrostatic interactions. So these electrostatic interactions actually keep the capsule in the vicinity of the bacteria, but it is not actually associated or covalently associated or linked to the cell wall. So that's why antibodies can bind to the capsule, but the phagocytosis is not going to be as efficient. So some of these GPSCs, like I said, are associated with single serotypes, but some are associated with more than one serotype, like serotype GPSC5. GPSC5 is also associated with drug-resistant strains. In case of antimicrobial resistance, there is 58% penicillin resistance, erythromycin is 29%, 13% clindamycin, and cotrimazole is about 94% with 53% in tetracycline. You can see here that there is some co-resistance as well, and penicillin and cotrimazole and tetracycline, although some numbers are different, which could be variability in the strain, that not all penicillin resistant strains may be resistant to cotrimazole. And we have done phenotypic and genotypic resistance. And in case of genotypic resistance, if you use meningitidal cutoffs, which are a little bit more stringent, then you see penicillin resistance rising in samples. So this is another good thing about whole genome sequencing, that you can actually compare the phenotypic and the genotypic resistance. You can also predict resistance by looking at your genotypes. Now, conclusions from PCV10: among 190 isolates, 66 GPSCs, 113 sequence types were isolated. Top 5 GPSCs were 5, 67, 25, 2, and 10, which were accounting for about 29% of isolates. GPSC 9, 10, 84, 650, and 43 were 100% MDR. And when I say 100%, it means that they were resistant to 4 or more antibiotics. Then GPSC 5, 67, and 2 had 40 to 80% resistance. The most important thing, or the highlight of our study, was we observed, or we reported, 2 capsule switching events. One was 19F, which is aligning now with 14, and 18A and 18C. So these were the 2 capsule switching events that we observed in our first cohort of samples. Then in PCV13 era, the serotype distribution among post-PCV13, you can see that 1 and 3 are not going anywhere. They are still persisting. Then there is 5, 19A, and all of these serotypes are present in PCV13. 19F is still hanging there. 18C, I just told you that there is a serotype switch. Then in carriage and disease, now these carriage numbers are really small, and you can almost ignore it, but it's good that we have them there, and we can show that they're also associated with non-vaccine serotypes. So most of the carriage now is with the non-vaccine serotypes. You don't find a lot of carriage with the vaccine serotypes, so which is again not a very good sign, because once you have carriage is the prelude to disease. So these are we need to keep an eye on these strains, because eventually these are the ones who are now going to cause an infection. The other thing is that, which is again the most important thing, is that we are still unable to eliminate serotype 1 and serotype 3, even though we have been vaccinating for the last 10 years or more. Serotypes by GPSC pre-post PCV13, so the overall situation remains the same, that some of the GPSCs are associated with more serotypes while the others are not associated. The ones that are highlighted red here are the ones that are present in PCV13. And then below here, you can see that each one of these, their antimicrobial resistance pattern, and penicillin resistance 78% in 43, 25, and 2. So 43, 10, 5, they are highly resistant serotypes. And then some of the serotypes, like serotype 2, is associated with single, not serotype, but GPSC2 is associated with a single serotype, and this serotype is serotype 1. And even though there is not a lot of resistance, most resistance is associated with 10, which is associated with multiple different serotypes. So overall, the pattern or the distribution of serotypes among GPSCs is kind of same, even though now there are different serotypes that are being represented in different GPSCs, but the overall pattern remains the same, that some GPSCs have only one serotype associated with it, and some GPSCs has more than one serotype associated with it. Some GPSCs have more drug-resistant strains associated with them, while the other GPSCs don't. So the conclusion of PCV13 overall, 37 serotypes, 35 GPSCs, 69 sequence types were detected in 120 pneumococcal isolates. Top 6 prevalent GPSCs were 43, 2, 12, 10, and the ones that are highlighted in green were also observed in post-PCV10. So these GPSCs are kind of very sincere GPSCs that they are still hanging out here. GPSC 10, 152, 67, and 9 have 100% MDR isolates. Then GPSC 43, 2, 12, 10, 26 have 72, 100% resistant isolates. So the overall general conclusions pretty much remain the same even after PCV13. We still have decent amount of infections are being reported in the hospital. We continue to get samples, but now they are shifting towards more non-vaccine serotypes. Now, if we look at the age-wise trends between 2017 and 2023, the first few bars are post-PCV10, then PCV13, I think, which started in 2021. You see that there is a still decent amount of infection. So 80% of infection, the orange bars are indicating adults, whereas the blue bars are indicating children. So regardless of number, we see infection in both children and in elderly. If you distribute this infection by invasive and non-invasive, in case of invasive infection, in 2019 and 2020, we only see infection in either children or in adults, but otherwise in 2023, we are still seeing invasive infections in both children and adult. In case of meningitis, again, we are seeing decent amount of meningitis. Most of the cases are still pneumonia, high rates of pneumonia in adults in both 2023 and in 2018. So what do we do now? How do we move forward? Or is there a way forward? So the next target that we thought might work is pneumococcal PSPA. Just going to give you a little bit of biology on pneumococcal PSPA. Again, it is a heterogeneous protein. It is antigenic in nature. But the important thing about PSPA is that it cross-protects. So one family of PSPA can cross-protect against another family. Now, most of the sequence is highly heterogeneous. It can be divided into alpha helical region, proline-rich region, and the choline binding region. The choline binding region is the region that actually tethers the protein to the surface choline or the cell wall. So cell wall has decent amount of choline or high concentration of phosphocholine, and that's where the choline binding region or PSPA uses its choline binding region to attach itself to the cell wall. The most important part of PSPA is the clade defining region. It's a very heterogeneous region, and based on the heterogeneity of this region, we can divide PSPA into 6 clades, which are then grouped into 3 families. Another important thing is that the clades don't respect serotypes. So if we look at it within the same serogroup, there are multiple different clades in these serogroups. Then you go into 19F, and these are the strains that are the ones that are causing most infections in post-vaccine era. And you see that each one of these serotypes have multiple different clades. So if you take 1,000, here they have taken 24 different strains of 6A, and all of these are representing these clades. Then 27 different strains of 6B, all of them have clades, clade distribution in them. So there, and the clade distribution is fairly different from each of these strains. Then in case of susceptibility to antibiotic, again, we are seeing a similar pattern that they are represented. And the good thing about this vast representation of these clades is that these clades are not changing as frequently as the serotypes are changing. So if we make a vaccine against PSPA, then because the antibodies from one PSPA or one family of PSPA cross-protect against the other family of PSPA, and 2, because they don't have any respect for serotype, there is a better chance that we might be able to stop this craziness that's happening because of the pneumococcal polysaccharide vaccine. So we did a little bit of our own study. We took about 20,000 isolates, and we looked at the clade distribution in 20,000 isolates, and we can see that the most predominant clade among these isolates is clade 1. then followed by clade 5, and then clade 4, 2, and a little bit of 6. And the interesting thing about 6 is that people have been working on the pneumococcal on PSPA diversity since 2000, or maybe even before that, 1998. And clade 6 had always been underrepresented, and clade 1 have always been overrepresented. So you see that yes, they are diversifying, but they're not diversifying themselves at the rate at which the capsular types are diversifying themselves. So we still have some hope. And these 20,000 isolates were collected or were taken from the GPS database. And we isolated PSPA from the whole genome by using specific primers. And then we did a lot of cleaning of that data. And after that, we started to assemble PSPA. And then we used some nested primers to identify clades within the PSPA sequences. So when you look at these little phylogenetic trees, you can see that the family 1, family 2, and family 3 can actually be clustered, except for few outliers. And these outliers are probably the new sequences, and we have not worked on these sequences extensively. And if we start working on these sequences, we might be able to assign them some families. But other than that, among those 20,000 strains that we have, there is a very good distribution of families and clades. In family 2, we have clade 3, 4, and 5. In family 1, it's 1 and 2. And family 3 is sort of by itself with only one clade, which is clade 6. And here you see clade distribution, very clean distribution of clade, where you have clade 1, 2, then 3, 4, and 5, which are all grouped in family 2. And then clade 6, which is kind of a loner, have always been by itself. Now, PSPA clades associate with invasive infection. So we did some odds ratios here, and it turns out that clade 1 has the highest odds of invasive infections, followed by clade 5. So now you know that if you incorporate clade 1 and clade 5 into that vaccine, there is a very good chance that you will be able to protect children against invasive infection. And then invasive potential of clades globally. So here we have strains from South Africa, then India, then Gambia, and you see that it remained persistent, that clade 1 actually is more invasive, followed by clade 5. The only exception is Peru. We had decent number of sequences from each one of these regions, and these predictions can actually or are more likely to have some weight. Then you can also associate clades with antibiotic resistance. And again, clade 1 seems to have the highest resistance against meropenem, followed by amoxicillin, erythromycin. But then there is a deep clade distribution, and clade 1, which is so here we are seeing a little bit of a different story. So more resistance is being shown in clade 2, followed by clade 1. Then of course, the other clades are trailing along. They are resistant. So just like the serotypes and GPSCs, where we can associate those GPSCs with resistance, you can also do the same thing with PSPA clade. So, there is a very strong association or this small protein, which is not going anywhere and is present on all pneumococci, can be used for discriminating between different bacteria. It can also be used for discriminating between invasive potential of a bacteria and carriage potential of the bacteria, and can also be used to discriminate against different antibiotics. Lastly, we wanted to see if there is any respect that these clades will show towards serotype. So here there is a low clade diversity was observed. So the clonal complexes and in sequence types, you can see that there is low clade diversity, but when you come to the serotypes, they don't respect serotypes. So there is very high diversity in clades. So the clades are, and I think I showed you in one of my previous slides also, where in one serotype, you can find multiple different clades. So this was the end of my show. But finally, I would just like to say one more thing that, there is still hope. Yes, we have a long way to go. We have lots of work that needs to be done, but it's not impossible. If you guys have any questions, I'll be more than happy to answer the questions.
Thank you very much, Dr. Shaper. And I think this was it's an amazing talk and probably one of the most comprehensive overview of pneumococcal biology that I've heard in a long while. So thank you for that. And I think we have time for maybe one or two questions if from the audience. Let's start. Tazeen, you have a question. Dr. Tazeen, you have a mic. Maybe I think you'll have to come here. Dr. Tazeen, you'll have to come here because so that
Hi, Assalamualaikum. So I'm Tazeen, I'm a clinical microbiologist, and I'm interested in knowing if you have identified any particular host factors or environmental factors that are associated with the capsule switching and the any particular groups that you can identify in which it is more prevalent, so you can target more expanded version of vaccination in those particular groups, covering more serotypes.
So, this is an amazing question. It's a very good question. And the reason why I'm going to enjoy answer this question is because I'm working with diabetes. Now, you see, when I showed my first slide where we were talking about the biology of pneumococci lives in the nose, right? So nose is an environment where you don't have a lot of nutrients, right? And the glucose that is normally found in the nose is 0. But when people have diabetes and their HbA1c and blood sugar starts to rise, the glucose in the nose is about 5 millimolars. And that change in the microenvironment of the niche is sufficient for pneumococci to do lots of things. We believe that this is a signal for pneumococci to transit. Just leave, move from the nose into the lungs. And that's why we think that people who have diabetes are so susceptible to having pneumonia, because when the bacteria arrives in the nose, it probably doesn't stay there. And if they are carried in the nose, they're going to immediately leave the nose. This is one way of looking at this thing. To answer your question, whether they switch their serotypes, it's an important thing to know. But for that, we will have to, and I think Dr. Stephen had done some work where they have done mathematical modeling of how capsular switching is occurring in the nose in polymicrobial communities, where they have shown that when they reside with different communities or with different bacteria, they do tend to change their serotypes. The frequency or the mathematical number that they have associated with it, which tells you how frequently this event is happening. I don't have it on top of my head, but I think it is a very high number. Whether environmental conditions or the changes in the environment of the microenvironment of the niche allow for this switching to happen, I don't know, but that doesn't mean that doesn't happen, because the change in this environment will definitely allow pneumococci to do something, either to hang in there or to move forward. So I'm sure that it is happening. What we need to do is we need to work on it, like continue to swab these people, because this is the easiest thing we can do. It's a natural environment. We are not, it's almost like an observational study, that we are not intervening. The environment is changing by itself, right? The other thing that we can do is maybe you need to come to Lahore. And then during the smog season, people have allergies, people have asthma, and that also changes the microenvironment of your niche. Okay? Because pneumococci is not present hanging in the environment. Unlike E. coli, where you can grow E. coli from the soil, you can collect E. coli from the fomites, pneumococci has to have a host. And unfortunately, human is the only host. The other thing that you can do is use mouse model. And geneticists are working on it. In fact, if you read Jeffrey Weiser's paper, he had worked on carriage. He's just the guru of carriage. So he had done carriage in human, like experimental carriage, which means that they had given bacteria to human, and the work was published in New England Journal of Medicine. And they have done a lot of carriage work in mice models also. And mice models are easy because you can change the conditions, right? You can make them diabetic, you can change their diet, you can give them smoke so that it replicates or duplicates environment where there is a lot of smoke. So yes, working in those environments will give you answers whether switching is occurring or not. But I am willing to bet my last dollar that yes, the switching is occurring. Good question, Dr. Tazeen. Thank you.
Thank you. A really interesting question and equally interesting answer. So I'm afraid we are a little over time, but I think maybe okay, maybe one more question. Naveed, yeah.
Assalamu alaikum, Dr. Naveed. Good to see you again.
Thank you so very much. So, Dr. Shaper, I have a very nice question, maybe. Since we have been actually using, the different vaccine and, against the most prevalent and deadly strains of the PCV, and we've been, seeing that the vaccine-covered serotypes are somehow, declining. But consequently, that is leading to, a niche, which is then, fulfilled by, the non-vaccine serotypes. And maybe, in most of the time, those non-vaccine serotypes are deadly too. So I'm curious, what are your thoughts on, is there any way, to come up or to abandon these serotype replacements, though it's a natural phenomenon?
It will be very difficult, Dr. Naveed, to eliminate the entire phenomena. Because when pneumococci lives in the nose, even though it looks like, or it appears that PCV7 serotypes are going down, but I'm sure that the little bit of PCV7 that is surviving in the nose, it's constantly exchanging genes with the non-vaccine serotypes. And that's how they are coming up with these novel new serotypes. So eliminating this mechanism is definitely very difficult. People have tried using lactobacilli, that, just give a shot of lactobacilli, because lactobacilli, when they start to colonize, they are going to produce a lot of gas, a lot of acid, and pneumococci doesn't have a way of fighting reactive oxygen species, so they are going to die out by themselves. But these things have really not worked. Pneumococci is hardier than we give him credit for. So I don't think that there is a way of eliminating this transition or transformation or constant evasion or constant evolution of pneumococci. But yes, changing the target of pneumococci might actually stop or prevent this phenomena for time being. And that's why I suggested PSPA.
Thank you. Thank you for the great answer.
Thank you, Dr. Shaper. I think it's interesting to note that when you talk about E. coli, salmonella, you're talking about one organism. But in the case of pneumococcus, when you say there are more than 100 plus serotypes, it is 100 plus organisms. So and that's why one way to do it is to do it in the form of increasing the valency of the vaccine, as Dr. Shaper also said. The most recent 20-valent conjugate vaccine by Pfizer, which has been approved for adult and but is not part of the expanded program of immunization here in Pakistan so far. And a theoretical and ideal vaccine would be a pan-serotype vaccine, which would cover all the serotypes. But of course, there's technological challenges and other difficulties in making that. So once again, Dr. Shaper, bahut shukriya. Thank you very much for giving this amazing talk and for joining us. So thank you.
Thank you. Allah Hafiz. Thanks a lot.